Hello everybody, my name is Bo. Today in this video, I'm going to review a research paper on virtual core flow cytometry. So what is flow cytometry? Flow cytometry is a technique used to detect and measure physical and chemical characteristics of a population of cells or particles. Traditionally, a sample containing numerous cells or particles is firstly suspended in a fluid and injected into the flow cytometry instrument. The sample is focused to ideally flow one cell at a time through a laser beam, and the light scattered is characteristic to the cells and their components. The scattered laser beam has two directions, the forward direction and lateral direction. The forward direction can give the information of the size of the cell or particle. The lateral scattering can give the information of the complexity or shape of the cell or particle. For more information, the cells are often labeled with fluorescent markers so that the light is first absorbed and then emitted in a band of wavelengths. Using this technique, numerous cells can be both quantitatively enumerated as well as classified by their shape or complexity or more information such as fluorescence. Flow cytometry has a lot of usage or application. Despite its application in cell counting and sorting, it can also be used to determine the cell characteristics and function. And also it can be used to detect microorganisms in all sorts of sources, such as ocean. For research-wise, it can be used in stem cell biology and oncology, and so on. A traditional problem of flow cytometry is that it uses a lot of sheath fluid in the experiments, during the experiments. So the problem of this research to address is that despite using a lot of sheath fluid to focus the cells in the scattering process, it developed another set of method to not use sheath fluid, but also concentrate the cells for detecting. So this research introduced a method to address the problem. Instead of using sheath liquid to position the particles with respect to the focused laser beam, it used a confocal microscopy to take a closer look at the area of interest. The setup of the system can be shown in the diagram down below. The scattering light coming from the source, which is the area of interest, is enlarged by a 50 times microscope and focused to a pinhole. Meanwhile, some of the focused light is reflected by a relay mirror and captured by a CSD camera. The CSD camera is used to monitor the image on the pinhole mirror, accurately align the laser, PSD, which is position sensitive detector, and the sample stream. The light coming from the focused microscope is then going through the pinhole and captured by three position sensitive detector, D1, D2, and FSC. As a result, the signals from the three PSDs can be analyzed to determine the position of the particles or cells in the confocal area. With this application, it can also be used to count the cells or particles. Also, part of the laser beam is also analyzed by two transducers. Two fluorochrome transducers detector is attached to this system to analyze its fluorescence information. To better compare the results of this flow cytometry with traditional cytometry, three experiments were done. So firstly, a simple test on the PSD is done to examine its capability to differentiate the different size and the position of the particle. Side-to-side motion of the beads of less than 25 micrometers for the two sides of the beads are done and measured to provide the information. On the left-hand side shows the side-to-side motion testing of the 1 micron meter beads. The measurement of 3 PSD is combined to determine which events come from the center of the stream and are suitable for a cytometric measurement. Scattered light from the center of the stream will passes through the center of the pinhole and listed as A. The light outside the virtual core will either enter only one of the detectors or will be detected by all three, which is shown in B and C. The expected result of trajectory of 1 micrometers and 6 micrometers is shown on the left-hand side of the picture on the right, and on the right-hand side of the picture on the right shows the real image of this flow cytometry results. As expected, the trajectory of 1 micrometers beats side-to-side motion is recorded as a ellipses or circle shape throughout the diagram. As can be expected, the 6 micrometer beat side-to-side motion will shown as smaller shape inside of the circle of 1 micrometer beats motion trajectory. The next two experiments was used to evaluate the efficacy of this flow cytometry. For this experiment, marine T cells labeled with CD4 and CD8 fluorescent monoclonal antibodies was used as source. As discussed before, the fluorescent scattering was assessed by the rest of two detectors. Picture A on the left shows the result from traditional flow cytometry. Picture B on the middle and picture C on the right shows the result from virtual core flow cytometry. Picture B in the middle is the result without applying a gate that determines the virtual core, and picture C is the result from virtual core flowmetry with the with the integration of the gate to determine the virtual core. As we can see, the virtual core cytometry, flow cytometry not only give the characteristics of both cells labeled with CD4 and CD8, but also give the quantitative information of those two. For further examining the efficacy of this flow cytometry, a raw seawater testing is done. This raw seawater has a microorganism called phytoplankton, which can be regarded as a scattering source. The phytoplankton is also labeled with fluorescent markers, and 1 and 6 micrometer beads is used to set up the gain and get better result. On the left-hand side is the result from traditional flow cytometry. On the right-hand side is the virtual core flow cytometry result. Again, the virtual core cytometry result reproduced both the general shape of the plot and can be used for enumeration and ratio concentration measurement of the particle or cell. So compare the results from this new flow cytometry with the traditional cytometry, which is the gold standard of cytometry. This new method presents a possible solution to address the problem of using a lot of liquid in the sheath, but also provide the same results which differentiate the size and the number of the cell particle. However, I think it needs a lot of calibration or calculation which is used to relate the size of their of the respective area to the number of the particle cells. So to draw a conclusion, this virtual core flow cytometry provides the same efficacy and efficiency as the traditional flow cytometry. Also, the system is less complex. It is easy to implement and reproduce the system. By removing the sheath fluid, not only the experimental cost was decreased, but also it provides a better characterization of the sample. Because in traditional flow cytometry, reanalysis of the sample is impractical because the sample is diluted 100-fold by sheath fluid with each successive analysis. In the future, as I mentioned, a correlation of the area shown on the diagram has to be done, in my opinion, to relate this area to the more precise results as the shape of the cells or particle or the numbers. Also, this system can be further implemented into more complex system, which where several lasers can be added to excite other fluorophores. That's all for my presentation for this research. Thank you for listening.